Hi, my name is Sohail. Today I am going to discuss the part 1 of prokaryotic transcription. It includes nucleoid, transcription bubble, template strand, initiation site, upstream and downstream nucleotides. Prokaryotes including bacteria and archaea are mostly single-celled organisms, and by definition they don't have membrane-bound nucleus and other membranous organelles. A bacterial chromosome is covalently closed circle, that is, unlike eukaryotic chromosome is not organized into histone protein. As we know, in eukaryotes chromosome is composed of DNA and histone protein. So, histone protein is absent in bacterial chromosome. The central region of the cell in which prokaryotic DNA resides, that is called nucleoid. Prokaryotes, they often have abundant plasmids which are extra chromosomal rings, which are shorter circular DNA molecules that may contain one or few genes. Plasmids can be transferred independently of bacterial chromosome during cell division and often carry traits such as antibiotic resistance. Transcription in prokaryotes as well as in eukaryotes requires DNA double helix, that is partially unwind in the region of mRNA synthesis, and the region of unwinding is called transcription bubble. Transcription always proceeds from the same DNA strand for each gene, which is called template strand. The mRNA product is complementary to the template strand and is almost identical to other DNA strand, that is called non-template strand. As we can see in this figure, this is the DNA strand which is called template strand, and the other one is called non-template strand. From this template strand, a complementary messenger RNA molecule is synthesized in the direction of 5' to 3', and this complementary messenger RNA more or less identical to non-template strand. The only difference is that in messenger RNA, all of thymine nucleotides, they are replaced with uracil nucleotides. So, in RNA DNA hybrid, adenine binds to uracil nucleotides in messenger RNA via 2 hydrogen bonds. So, there are 2 hydrogen bonds between adenine and uracil nucleotides and 3 hydrogen bonds between guanine and cytosine. The nucleotide pair in the DNA double helix that corresponds to the site from which first 5' messenger RNA nucleotide is transcribed, that is called +1 site or initiation site. The nucleotides preceding the initiation site are given negative numbers and are designated as upstream nucleotides. On the other hand, nucleotides following the initiation site are denoted with positive numbers and they are called downstream nucleotides. To sum up, prokaryotes, they do not have membrane bound nucleus and other membranous organelles. Bacterial chromosome is directly present in the cytoplasm and do not have histone protein. The central region where bacterial chromosome is present, that is called nucleoid. Extra chromosomal rings are called plasmids. They contain genes for antibiotic resistance and they are directly present in the cytoplasm. The region of DNA where transcription takes place, that is called transcription bubble, and transcription proceeds from the template strand of DNA and a complementary messenger RNA molecule is synthesized in the direction of 5' to 3'. One of the difference between RNA and DNA is that thymine nucleotide is present in the DNA and uracil nucleotide is present in messenger RNA. mRNA molecule is synthesized from 5' to 3', and the nucleotide pair in the DNA where transcription starts, that is called +1 site, that is also known as initiation site. Nucleotides preceding the initiation site are called upstream nucleotides and the nucleotides following the initiation site are called downstream nucleotides.